welcome to the Vernal Pool Phenology Project informational meeting for 2024. Just to give you a little brief overview of the agenda for the meeting, we're just going to give a brief overview of the project. We have participants that are attending this session that participated in this project last year, but we also have participants participants that are potentially going to be joining this year. So we've kind of got a mixture. So we're going to try to talk to both audiences, and we're going to keep it a little bit briefer because last year we still have the recording from the informational session from last year. We'll go over briefly what you would need to participate in this project, sampling protocols, and with an emphasis of some updates and clarifications for the 2024 protocol, and then a brief introduction of some of the teaching materials that are provided. And then pretty much at the end, we're going to have an open time for questions and answers from anyone. You'll be able to, along the way, feel free to put questions in on the chat. We'll be monitoring that as well. But then we have that open question and answer period at the end. This session will be recorded and posted to our YouTube page, along with previous webinar recordings. Next slide. So briefly, the Vernal Pool Phenology Project is a project from the Ecological Researches Education Network. What is AREN? AREN is a network of faculty that teach at primarily undergraduate institutions or other teaching or have teaching intensive faculty positions at other institutions, where we welcome, where we're trying to incorporate research, authentic research into undergraduate curriculum, both teaching and through independent studies. But AREN, so that's how these research projects are designed. They're designed so that we're able to incorporate this into the classroom and with undergraduate students. However, many of the AREN projects, as well as the VPP project, welcomes participation from a broad audience. So we already have had environmental educators, citizens, a variety of people that are participating in this project. So we welcome that. As an Ecological Research as Education Network project, again, we're emphasizing collaborative research instead of just having teaching and research experiences local. This allows us to broaden our reach so we can have collaborative research, conducting relevant research questions at regional and continental scales, and with a focus of producing high quality publishable data involving undergraduates. We have the 4 co-leaders of the Vernal Pool Phenology Project. I am Mary Beth Kolishvari from Siena College. Will you guys introduce yourselves?
Sure. I'm Dave Steinberg at the University of New Hampshire.
I'm Jennifer Pernhage, also at University of New Hampshire.
Tilina Surasinghe, Bridgewater State University, Massachusetts.
Next slide. On the Ecological Researches Education Network, we have a homepage that addresses the focus of the project, amphibian and plant phenology at vernal pools. And the overall broad question that we're trying to address with this research is, how do local and regional factors affect spring phenology of pool breeding amphibians and trees? So we're looking at vernal pool ecosystems as a whole. So we're looking at the pools, the hydrology, the faunal species, in particular pool breeding amphibians that breed at these pools, and the surrounding vegetation. So how does climate change potentially affect all these components of vernal pool ecosystems? So climate change has led to changes in phenology or timing of life history events in plants and animals. And so we're looking at, are all these animals and plants, the leaf budding out, the pool filling and drying, are all these tracking as climate changes at the same time synchronously, or are there mismatches, potential mismatches as a result of that? In order to address these factors, which is why we're trying to we're not going to be able to address these types of questions looking at just one site or a couple of sites. We're trying to broaden our reach across a large scale, which we've been successfully doing with this project. So we'll be able to tease apart local versus regional factors, looking at asynchronicity between plants and animals, as well as vernal pool hydrology over time. And with the ultimate goals to share our research findings in conference presentations, manuscripts, etc. Next slide. And I think that's you, Dave, is the next one.
It is. So as project participants or potential participants, you'll be responsible for monitoring the phenology of amphibians and trees at your own local vernal pools, at least across one field season, but we hope that you'd be able to participate for multiple years. Next slide, please, Jen. We really want to make it easy for folks to participate in this project. And to do that, we've developed a number of materials that are available on the Erin website and on our Google Drive that should hopefully make it easy for folks to jump in. But you'll see on the next slide here that you are going to still need some things in order to participate. Most critically, you need some sort of pool or vernal pool. This needs to be forested, fishless, and less than 0.2 hectares. If you have any questions about your potential pools, please just send us an email and we can walk you through that as well. Next, you'll need about $600 to $700 worth of equipment. We know that that number is quite large. Our hope is that actually many folks may have a number of these supplies sitting around in their storage sheds or labs or classrooms, like waders and binoculars. But there are a few sort of specialized pieces of equipment that you may need to purchase specifically for this project. And again, we have all of those listed here on this slide, but also on our website, and we have URLs, hyperlinks that you can just click on to get to a place where you can purchase those. Now, in the bottom image, you see some acoustic recorders there, and we're using that to monitor the calling behavior of wood frogs at our vernal pools. Specifically, we're encouraging folks to use Audio Moth brand acoustic recorders. And if Jen clicks the next button here, you'll see that we actually do have some recorders available to lend out to folks. So if for whatever reason, financial or otherwise, you're unable to secure an Audio Moth or another acoustic recorder, please send us an email and hopefully we'll be able to get some equipment out to you so you can participate. Next slide, please, Jen. Thanks. You'll also need to be able to complete twice weekly surveys for 6 weeks of this project. Those 6 weeks are gonna be around early spring. So when our wood frogs and spotted salamanders come out and start breeding, you'll try to start doing a survey about 2 weeks before you might expect these amphibians to start emerging. So it's a little bit of guesswork, but again, we can help you try to determine maybe an appropriate starting time for your specific location if you reach out to us offline. We'd love for you to participate year after year after year, but we're asking participants to collect data and share data for at least 1 year in order to hop on to our presentations and any publications that may emerge from this. Next slide. Also, you need to have a willingness to share data. We do have specific policies in place for data sharing. So it's not like you're sharing it with us and then we're putting it out into the world where anybody can use it. Specifically, we're going to be using it to answer questions that Mary Beth posed a slide ago. So just being willing to collect your data and then upload it to the site so we can all work on it collaboratively is important. Next slide. Now, the really important thing I think for this presentation is that we have made some changes to our protocol based on feedback that we received either via email or via Qualtrics survey or Google Form survey from participants last year. Those are sometimes changes are in the form of clarification. Sometimes it's actually a straight up change. And so before Thelina gives you a brief rundown of the methodology, I do want to just say off the bat the 5 major changes that you're going to be encountering as you move through our modified protocol for spring 2024. The very first thing is we tried to clarify our tree mapping protocol, how we select trees to monitor over the course of our 6-week season. There was a little bit of confusion, and we hope we've solved that problem. If we haven't, please reach out to us and let us know. Also, in our previous protocol, we had asked to have hobo data loggers in 2 different locations, the deepest part of the pool and the center of the pool. We've now changed that so that all hobo data loggers will go on the same water gauge at the deepest part of the pool. Hopefully that makes things easier. We're adding 1 new phenophase for our tree phenology. Last year we had bud break, leaf bud break, emergence of leaves, and flower emergence. This year we're also adding swollen flower buds as an additional phenophase that we'll be able to use moving forward. We're asking folks to download data or remove their hobo data loggers on September 1st. That's a bit of a change from last year. This is just so people don't feel like they need to rush out to the pools right after the end of the spring amphibian breeding season. It also allows us to capture a little more temperature data. And then finally, we have 1 team leader designated as our email correspondent. And we hope that this will streamline communication so we can get responses out to folks in a more timely manner. Next slide. And I believe we're on to Thelina.
Thanks, Dave. So let's talk about very briefly about the methods, right? Like Dave mentioned before, this is going to be, I'm going to keep it very brief because you can always refer back to the recorded video from the previous season or consult our protocol. So in terms of site selection, you're looking at, you look for a pond that is fishless. That's really important criteria. That's, smaller in size, and, should hold water either seasonally or permanently. So as long as it is fishless, permanent water is fine. And should be associated with forest, not grasslands or other type of habitats. We should have one or both of the focal species, the wood frogs or the spotted salamander. If you have both, that's great. And in terms of the tree selection for the tree phenology component, you should have at least 1 species of a woody tree with at least 5 individual trees within a 5-meter buffer. And recall that if you do not meet the minimum criteria within the 5-meter buffer around the pool, then you expand to a 10-meter buffer. And then we strongly recommend that you start the site selection in fall, although the data collection happens in spring. Next slide, guys. And we have provided the protocol which actually goes into detail about how to measure and how to create the instrumentation for this particular purpose. So measuring hydrologic variability, especially the depth, is an extremely important thing. So we actually, provided details about how to create this homemade water gauge, which actually you construct with a PVC calibrated, hand calibrated PVC pipe that's anchored in place based on a rebar that is driven into the substrate. And then, of course, the calibration can happen with a permanent marker, right? Or with electric tapes, just to make sure that those things don't disappear. And you make measurements of the water level during each time you visit the site for other phenological observation throughout the 6-week period. And then attached to this water depth gauge. You will have those hobo pendants that measure both air temperature and light levels. So one of the hobos will be ambient on the top of the PVC depth gauge, and one submerged in water that measures the water temperature. And then canopy cover, which only you measure. So the pendants, the hobo pendants, you deploy them, right? And then you recover them on September 1st. And by that time, some of your ponds may not be dry, but that's fine. You still collect it, right? If ponds are dry, yeah, definitely that's actually marks the end of that particular pond phenology. But even if it is filled with water, you collect it by September 1st. The canopy cover measurements that are taken by your smartphones by just taking just a regular photograph. It's really important that you take that photograph at the same point in the pond repeatedly. And it's also important that you properly hold the phone parallel to the straight. And you take two photographs. One is actually, when it is leaf off, right? Fully leaf off, and then fully leaf out. So roughly, it's depending on, the leaf phenology in your site, so you can determine what is the best time to take those two photographs for a given year. Last slide. Well, what is a specific protocol for phenological monitoring? There are two parts of phenology. One is the plant phenology, other is the amphibian phenology. For the plant phenology part, well, when you decide to start, well, again, this is when you need to know your local knowledge, right? So you first determine this based on the general amphibian activity, right? When do you hear the first wood frog calling, right? And then you start you try to, when you get the general idea when the first wood frog is calling, generally in your local area, your region, then of course plan a little ahead of that time to start monitoring, because so that in this way you don't miss the first date. So now back to the plant phenology, specifics about the plant phenology. You have 1 to 5 tree species that you picked based on the dominance within that particular buffer we mentioned earlier on, and then you start monitoring the phenology for a period of 6 weeks, right? And then you measure, you do this twice a week within that 6 weeks period. And the phenophases are leaf bud break, or the emergence of needles, depending on whether you are looking at deciduous or evergreens, young needle emergence, and or the young leaf emergence. That's second phenophase. Third one, this is a new one. Dave referred to this already. We are looking at two flower phenologies. One is swollen, like big fat flower bud, presence of that. And the second flowering one is actually completely like open flower. And so even if you don't observe any of those, targeted phenophases, it's really important that you record that, right? And in terms of the amphibian phenology, you are looking for, either both or one of the two focal species. And what we are looking at is first appearance of egg mass, as well as the first appearance of spermatophores. And there's another amphibian phenophase that we are interested in, that is the first wood frog call. We do that by deploying an audio moth. Now remember, the audio moth deployment should also start before the first date that you guess based on your knowledge, or you can actually consult your local experts in the discipline. And also you can refer citizen science websites like Frogwatch or a National Phenological Network in order to ascertain how early you should start in your region. I think that's all three in my quarter.
Yep. So we just wanted to mention quickly a few things about educational teaching resources. We won't go into all the details because, again, we have previous recordings and you can review all the materials, and we're happy to talk with you one-on-one about them. But we wanted to mention that we do provide some teaching materials for this project and probably will add more over time. The teaching materials and just the project in general is very adaptable. We've used this for all sorts of different types of classes and teaching opportunities, including those listed here. And we also have in the past 2 years hosted an kickoff meeting. And we got feedback that some people would like us to host it earlier than we did in the past, because their field seasons start earlier than we were thinking about up here in the north. And so the kickoff meeting is really it's kind of like a pep rally. It's just meant to get all of the students and all of the faculty, all of the people who are participating in the project on a call together to realize that they're part of something bigger than what's going on just in their class or at their institution or at their location. So it's absolutely not required, but it's just something that we like to do because we've recognized that our students really enjoy that idea that they're not just doing a lab for class, but that they're part of a bigger, real, scientific project. And so we'll send more information about an annual kickoff meeting soon. And then this is just a screen capture of the first page of our teaching document that shows kind of some of what's included there. So a little bit about includes actually links to some of the materials that folks have mentioned already today, some of the kind of expected effort and general timeline, equipment and materials, and then has like a pre-lab handout that you can adapt, some worksheets or handouts for setting up your study site if you want to do that part with students, handouts for kind of a lab for going and doing the surveys. Of course, we have the data sheets that you're going to need to do the surveys. And we also provide some information about how to identify different types of egg masses if this isn't your area, if it's not something you and your students are familiar with. So being able to distinguish wood frogs from spotted salamanders and spotted salamanders from blue spotted salamanders and things like that. And then some plant phenology kind of cheat sheet information. The one caveat I'll share about that is that we can't share, obviously, tree phenology information that is relevant to everyone in the project because everyone has different trees that they're monitoring. And so that's something that you're definitely going to have to adapt a little bit more for your local site. And then we just wanted to mention that there's so much more that you can use these sites for once you set them up. And you now have this phenology data and kind of some acoustic data, this monitoring data at these vernal pools. We've set up trail cameras and we do kind of just broader surveys, reading the landscape type surveys with our students. None of that's required for the Vernal Pool Phenology Project, but once you've set up these study sites, these areas, you can do so much more with other classes and with your students at them, including independent projects, honors theses, things like that. So there's a lot that we have done already that our participants have done and a lot that you could do. So yeah. And then this is a map of our current, as of spring 2023, sites, so participants from 2023, or this might be a few more than who actually participated, but we had about 30 participating institutions last year, and we're hoping to have more. We're hoping that if some of you didn't participate in the past, that you join us this year. And we wanted to stop telling you stuff at this point and start answering your questions. And so we'll just open it up for questions. I know there was one question that I saw in the chat that maybe we want to address. I know Mary Beth was kind of addressing it a little bit, but there was a question about the water depth gauge that you're installing. And the question specifically is, are we measuring water depth from the top of the muck, in quotes, or are we measuring water depth from the bedrock impervious bottom, or as far as you can hammer the gauge down? And that's a good question. I don't know if anyone wants to address that. Like, we've talked about that a lot, that can be tricky and it can also change from year to year sometimes, like where the muck is. So does anyone, Mary Beth, I don't know if you wanted to elaborate on some of the stuff you put in the chat.
Yeah, I guess I can just say there's an art to figuring out where to settle the bottom of the gauge. You do the best you can. think about it, what you're trying to do is when that pool dries, where's the bottom of the pool going to be? And yeah, you don't really know, but you do the best you can. And believe it or not, I mean, in Acadia National Park, we had 72 of these things. We did a great job of figuring out where that was. So, yeah. Does that address the question?
Yeah, I mean, I think it's probably the truth is it's somewhere between the impervious bottom and the muck, because the muck will like lose some of that water in it and settle down. And so it's like somewhere in the middle. And there's going to be some margin of error there. But yeah, like Mary Beth said, you do the best you can, but it's somewhere between the two limits that you mentioned, the impervious bottom and the top of the muck.
Yeah, and I do want to mention that, sure, there is an error in that measurement, but odds are that error is consistent, right?
Sure.
Across, because it's likely the same person or trained by the same person is measuring it. So I think, just feeling your way where you are hitting that, the perfect spot on the substrate is actually the best way to go.
So let us know if you have more like follow-up questions about my pool does not dry up. So somewhere in between. Yes.
Yeah.
Yeah.
Anchor it there and you're going to be consistent. It's
exactly. Good. Perfect. And if you want to talk about it further, like they're always, everybody's site is unique in many ways. And so if you ever have questions and want to talk about it further, one of us is more than happy to have that conversation. But yeah, are there any, I didn't see any other questions yet. Are there any other questions that people have? We have everything except the sensors for the project. So doing a quick ask if anyone has extra hobos in Northern Virginia. Okay. We can check and see if any of us have any extras for one site. So you need 2.
Hi, yeah, that's me.
Very good.
Yes, ideally too. We have lots of pools on site, but we have one really good pool. And I'm actually was wondering, like, it's probably 15 or 20 centimeters deep at its deepest. And right at the deepest point is where there will be like piles and piles of egg masses, literally piles of them.
Yeah.
And so I'm just wondering, I guess that will be something we might have to deal with, is like egg masses obscuring the top of a button. I don't really know what we do about that, but
that's okay, because if I understand what you're saying, and correct me if I'm not, the one that's in the water is literally submerged underwater. So it doesn't need, it's okay if it's obscured. I mean, it just needs to be getting the water temperature. The one that's on the very top of the water gauge, the hobo that's on the very top, obviously that's not going to be an issue. So does that
gotcha.
make
Okay.
sense?
Yes,
Yeah.
copy that. Okay, yeah.
Yeah. And are you able to, it sounds like you are, but are you able to see those egg masses, even though they're in the deepest part, from doing perimeter surveys, like without wading in?
Yeah, we actually, a tree fell over the deepest
Oh,
part of the
nice.
pool last year, so we kind of shimmy out on it to check when
Perfect.
no kids are there.
Yeah, that's great. Yeah,
Yeah,
exactly.
it's good.
And because we've gotten this question about egg mass surveys, and whether we should be wading in. We've had long conversations about it, and we've opted to just continue doing perimeter surveys for the most part, like to not require people to wade in to do surveys. But all you're going for is, and I know this wasn't your question, but it just inspired me to think about this. All we're going for is like the first egg mass deposition, right? And so if you're missing some egg masses, like that's okay. You don't have to do a count, I guess is what I'm saying. So I just wanted to clarify that for everybody.
I want to add something once you're done talking.
Yeah, go for it. I was just looking at the chat. Go for it.
Yeah, so about the hobo data loggers. So there is a new version of the hobo light temperature meter, and those in the new protocols, it identifies both the previous ones we used or that. So those are fine to use as well.
Yeah, and one thing that we don't want to go too far in the weeds, but we will mention, since you mentioned that there are new versions, I will say if you're purchasing the new version of the hobo pendants, we really strongly encourage that you purchase the temperature light version, not the temperature only version. even for the one that you're putting underwater. And the reason is that, as far as we can tell, the brand new version of the Hobo pendants that is temp only, the batteries can't be replaced. So that makes them essentially disposable after the batteries die. So if you're using the new ones, go for the temperature light ones. And we clarified that in the protocol as well. All right, we have another question, which is, I would like to hear some stories about how well this went with students. Any student issues to expect and favorite moments? Love that question. And then we have, if there are no wood frogs or spotted salamanders on the island, is it possible? Oh, like to, I guess, if it's Alexander, if you could clarify, are you asking, could you participate but not do amphibian monitoring and only do trees? So if you could clarify that in the chat, that would be great. And maybe we can address Christine's question about stories and maybe unexpected issues and favorite moments. Does anyone have anything off the top of their head?
Yeah, I mean, I think I'll just say that we all incorporated it differently into our classes or not our classes. So I, for example, I had an independent study student working with me. Jen and Dave worked collaboratively with their labs where they monitored over a couple weeks. So we all encountered different types of experiences as a result of that. But that's one thing about this. So I know from my perspective, it was great to have an independent study student or a couple students working on that, because then I could work very, close hand on, hand on hand. I don't know what I was trying to say. Face to face, like hands on with a student in terms of being able to train them and identify them, and in terms of making sure you got the quality control on the monitoring. Jen or Dave, maybe you could speak about having classes of them, because you guys had a very different experience.
Yeah,
Like, way to incorporate it is what I meant. Yeah.
Yeah. We incorporated the surveying and even site setup and tree mapping and all of the aspects of the project at various times into different courses. And we would have, like, some classes would monitor for a couple weeks and another class would come and monitor for another couple of weeks. And we would have classes of up to, 30 people out there at a time. And we would just divide them up on different activities and have them do different aspects of the protocol. And as long as you're organized and do a little bit of training, first, it went really well. The students were very excited I was actually okay, so Dave and I are kind of come more from the amphibian side of this project, introducing this to our students, and I was really thrilled with how into learning the different tree phenology, like looking for the different tree phenophases, our students ended up getting, even though they're like, a lot of them are like wildlife students who are more into the frogs and salamanders. So it was really, really fun. The students have fun, some of them get to wade in. I don't know, it's a blast. Like, I mean, all of the different classes that we did this with had a great time. And we also had a couple of students get so excited that they did an honors thesis project or an independent study, and got to present that at undergraduate research conferences. And so it's been really productive so far. We got Dave, unexpected joy, a couple of students who got wildly into, yeah, that's what I was just saying, into monitoring tree phenology. It was like such a fun surprise to see how excited they got when they saw like a bud or like a leaf emerging. It was really cool.
Well, it's also pretty interesting. It really helps with their observation skills. So for example, around here, my God, it seemed like it was gonna go like a couple weeks earlier than ever. Everything was warming up, blah, blah, and some of the other species were starting to looked like they were about to bud burst. Yeah, that's it. Anyways, and then it got cold for weeks. I mean, it's just like, it's being able to get them to be tuning into things like temperature and watching it and saying and seeing these pauses. It's so interesting.
I agree. And I think I love what you just said, Mary Beth, because that was a real thrill was seeing them cue in more on their observational skills and email us outside of class about the fact that they were noticing the different phenophases that we were asking them to look at our vernal pools at other ponds on campus while they were walking around, and like sending us pictures of trees like, oh, look, the red maple's, flowering here, but not at our sites yet. And noticing that the wood frogs were, depositing eggs in the open ponds on campus, but not in our vernal pools. So just seeing that it extended beyond the classroom or beyond the vernal pools, and to just them walking around campus was very, very cool.
And I'll just mention another thing, that when we were beginning to develop the protocols for this project, we had one person that had done plant phenology measurements before on campus, and she's like, oh, and she's a plant person, and she's like, oh, when the leaves, the buds burst, they all kind of go at the same time. Well, that's not what we found. Individual trees are very different when you're thinking about in a forested situation. So it was pretty interesting as well, and enlightening, and it sure makes sense, but
Yeah.
And I
Yeah.
kind of feel like, just kind of thinking about the phenological asynchrony was also quite new. Like from the theoretical point, it's so cool to think about, these, deep in the phylogeny, looking into two different taxa and trying to theorize the phenological asynchrony and the underpinning mechanism behind it. I thought that was cool, really interesting too. And students found that, like that level of thinking interesting.
And by the way, I mean, I don't know how many other people are doing this. I know we have some participants who aren't working at universities, but Dave and I and our local collaborators here brought this project to some of our K-12 schools in the area. And we had third graders that were like telling us their hypotheses about the project, that we were like, wow, we didn't even think of that hypothesis. Like, I mean, it was really amazing to see what some of these like 8-year-olds are telling you they think about your project, and you're like, that's a great point. So it really works across all ages, and that was a real blast as well. We do have.
I'm just going to say one more thing.
Yeah,
We've
yeah.
really gotten off on that question there, but I just want to mention that I've also, been able to bring this more into my classes and speaking with students. And I had a summer research student. She was so awesome. We had some extra time. I'm like, hey, let's make a phenology trail. So now we've got a phenology trail on campus. And we've also, it's a really good selling point in terms of like, hey, people understand, I've heard about climate change. I think they get things about the glaciers and the warming temperatures and things like that. But how does it affect biological communities? And so by getting them to think about this and seeing how scientists study, we've gotten a lot of mileage on campus with that. So it's been great.
Yeah, that was a great question. And we could probably keep going forever.
I guess we'll stop on that.
Oh, there's more in the chat. Other people are putting great things in the chat. So that's why I love it. But I do want to make sure that we address some of the like logistical
protocol
questions
questions
that are coming up. Because that's important. So I want to make sure I don't miss them.
Yeah.
Aaron had asked, I'm still waiting for a follow-up from Alexander, I think it is. And then Aaron had asked, we are wondering how we should deal with anomalies in phenology first, and the idea that like, and I think Mary Beth might have jumped on this a little bit accidentally in answering the last question, but for example, just a few leaf buds burst a week before the majority. And then similarly with amphibians, you can have multiple waves of eggs being laid. We currently are the minimum, because we're trying not to overburden, our participants. We are currently going for the firsts, like the absolute firsts. So the first, I think I'm trying to remember off the top of my head without looking at the protocol. I think it's like 1 to 3. It can even be like just a few buds bursting, right? Counts as that phenophase being seen. It's not like you have to have 30% of the tree, right? Mary Beth and Selena and Dave?
Correct. Although I think some of our collaborators, I think Lori has been doing more detailed work.
Which is great.
But then that's just for her own project with her students.
Correct.
But
yeah,
for the larger scale project, we're not doing that.
Yeah, like the bare minimum is
whatever.
just that first.
Yep.
That first wood frog egg, that first spotted salamander egg mass, that for all of that, the firsts. So hopefully that answers that question, Aaron.
Jen, there was a question about that Nantucket Island. Probably, I don't know whether we addressed that.
I asked it, but then I asked for a follow-up because I wasn't entirely sure what the question was. Like
Well, I think
wasn't-
it's like if they don't have wood frogs or spotted salamanders, can
Yeah.
this be part of this study? So that is what we've required, having at least one of them known to breed at that site.
Yeah.
I mean, you
So-
could run into a year where you have very few, or you don't have it present that year, but you know that they breed there.
yeah, and I just want to
Yeah,
make sure that's what the question was asking. So
Right. That was a question. I believe that came up before through email. I do recall responding because I'm from Massachusetts. So so my it sounds like a joke, but seriously, like come over to the mainland. We have plenty of both. So unfortunately, biogeographically speaking, in the islands you don't have it. So I believe that disqualifies because biogeographically you don't have it. It is not even once in a while, right? Like, historically, we don't have either of those species in the Massachusetts islands. I'm not aware of any of those being in the islands.
I would encourage you to feel free to use this project and these protocols and focus on fairy shrimp. But like, for example, we're not having people wade through pools.
No.
We're not focusing on fairy shrimp because of all the problems with on not detecting them when they're actually there.
yeah, and you could still use our plant phenology
Yep.
protocols if you'd like to, and all of that, and whatever teaching materials are useful. There was a follow-up on our first question, like what we meant by that, and I just wanted to address that. So related question, are we only interested in first or abundance? And I think I mentioned that kind of casually, but wanted to articulate it. So we're not doing abundance, although we tend to write notes on our data sheets to be like, oh, we saw more. But really, we're just doing that first. That is the baseline data point that we're looking for. I mean, the threshold data point that we're looking for.
I've had a great question.
You found a great question?
Yeah, what is
Okay.
your timeline goal for having data
Yep.
submitted that was collected in 2023? I love it. Yeah, well, our goal was to have it submitted last summer, but we didn't get you the information. So you couldn't do it. Anybody got a good answer for that? I haven't uploaded my data yet.
Maybe instead of us just like throwing something out right now casually, it's something we should come up with like a hard date that we really want it done by, and we'll send that in our follow-up email. But it's a good question. We're glad you asked it.
Yeah,
Yeah.
it's not something we have forgotten. We have been talking about that, but like
Sure.
getting things our group ready for the new season is actually came up so fast. And then once the season starts, like definitely we will jump on that one and set a hard deadline on when to upload that data. And yeah, please do send any questions you have regarding that. And we apologize for the delay in communicating with you guys on that one.
But hopefully
But
everyone
we have
has
had
their
people successfully upload their data and
Yeah.
they followed the instructions and they gave us a thumbs up. So that's very
Yeah.
promising. And this will give us a great opportunity to be able to look at, what was our preliminary data from last year and what kind of patterns we have. So it's going to be very exciting.
Yes. I think we've addressed all the questions that have been asked in the chat so far. Does anyone have anything else? You can also unmute yourself and ask the question if you'd like to ask it or put anything else in the chat.
I'll mention one other thing. We've got at New York State Parks, we had an educator piloting this last year. She plans to do classroom, field exercises with school groups, etc. And now we've got another state park that they just put up when they've got a hole right outside their their nature center. They have a vernal pool right there and a boardwalk that goes right near it. So they're going to be incorporating that into their lesson plan. So we've got a really wonderful reach with this project, and it's something that is being, and I'm going to encourage them to help maybe work on developing materials maybe targeted towards nature centers to be able to share as well, because ours we're probably more focused on more undergraduates and that kind of work. So there might be some other types of ways of incorporating it and sharing that information.
I'll just say, I think I may have put a comment about this in the chat during our presentation, but we have updated the protocol and just have a few kind of cosmetic final edits to accept before we share the link. But in our follow-up email after this call, where we share a link to the recording of the call, we will share a link to the 2024 project materials, including all of the equipment lists and teaching, materials and the updated protocols. In the email announcing this meeting, we did share last year's information, which is largely the same. So it can give you, if you're trying to plan for the semester, it can give you a good indication of like what to base that planning on. But we'll share the like polished, updated stuff very, very soon, like later this week. Any other last questions or I mean, we can stay on for a bit longer if people do have questions. I'm not trying to rush, but any other questions or thoughts from leaders?
Just excited that there are so many people on here that are also excited. It's
Yeah.
great.
Here, here.
Great. Well, maybe we'll just stay on in case people have lingering questions. But otherwise, call it. So we're not taking up people's precious time in January. So thank you for coming on such relatively short notice. We appreciate it. And we'll share the recording so you can share it with other people or refer back to it. And we'll follow up with an email very soon. Thank you.
Thank you.